Aerobic respiration is the process of producing cellular energy involving oxygen. Cells break down food in the mitochondria in a long multi-step process that produces roughly 36 ATP.
Glycolysis occurs in the cytosol and breaks down glucose into intermediate steps. This requires 2 ATPs, which enzymes will dephosphorylate into ADPs at steps 1 and 3. Phosphofructokinase at step 3 regulates respiration by being inhibited by an overproduction of ATP and citrate. Glucose is split into glyceraldehyde 3-phosphate and an isomer that later converts into G3P. After splitting, each G3P has 1 NADH produced, 2 ATP produced, and finally 1 pyruvate produced, which will enter the mitochondria.
The Krebs cycle oxidizes organic fuel derived from pyruvate. First, acetyl-CoA from the pyruvate adds its 2-carbon acetyl group to oxaloacetate, producing citrate. Another CO2 molecule is lost and the resulting compound is oxidized, reducing NAD+ to NADH. The remaining molecule is then attached to the coenzyme A. 2 hydrogens are transferred to FAD, forming FADH2. And oxidizing succinate. In addition, a water molecule rearranges the bonds in the substrate and malate is formed. Then the substrate is oxidized, reducing NAD+ to NADH, and then oxaloacetate is regenerated.
The electron transport chain is the final part of aerobic respiration. The ETC is located in the bilayer and is made of 4 multiprotein complexes and 2 freely moving electron shuttles known as Q and cytochrome C. The ETC begins with the transfer of electrons from NADH to complex 1 and the movement of H+ across the membrane. The electrons then follow the chain, following closer to the nucleus of progressively more electronegative elements in each step, eventually binding to O2 in the matrix to form water at the end of the chain. The ETC pairs the exergonic process of lowering the potential energy of electrons with the endergonic process of ATP synthesis via ATP synthase, which uses the potential energy of concentrated H+ ions to make ATP from ADP and inorganic phosphate.